You are listening to the Synapse Performance Podcast with me, David Nolan, and today I'm joined by lecturer, researcher and strength coach, Dr. Cody Haun. Hello, and you are welcome today to episode 82 of the Synapse Performance Podcast. I am your host, as always, David Nolan. If you're a new listener to the podcast today, firstly, welcome. I hope you really enjoy what we do here. If you're a returning listener, I thank you again for your loyalty in downloading and supporting the show each week. Either way, make sure you're subscribed to the podcast on iTunes, Spotify, or wherever you listen so you can stay up to date with each new episode. And if you enjoy what you hear today, maybe head over to iTunes and leave us a rating and review there. It really helps us just grow the show and get it out to a wider audience. This episode, as I'll do with all the ones in the future going forward, will be available on YouTube also, so maybe head over there and subscribe to the YouTube channel. If you want to follow me personally on social media or Synapse, you can do so at Synapse Performance on Instagram, David_Synapse on Twitter, and then David Nolan or Synapse Performance on Facebook. And if you ever want to email me with any questions or inquiries about our services here, you can do so at david@synapseperformance.ie. So, on to today's guest. It's a pleasure to once again bring Dr. Cody Haun back onto the podcast. Cody is a lecturer and researcher at Auburn University, as well as being a strength coach with Stronger by Science, ran by Greg Nuckols. So today we get a bit nerdy in our conversation around muscle hypertrophy. We delve into kind of the deep science around the mechanisms of muscle growth and whether we can bias through our training, whether the muscle growth is myofibrillar or sarcoplasmic hypertrophy. So a bit technical, a bit deep today, but I hope we make it as accessible and enjoyable as we can. Hope you really enjoyed today's episode. If you do, maybe you'd consider sharing it on your social media or sharing it with a friend or colleague who you think would enjoy the show also. So let's get Cody Haun onto the show. It is a pleasure to once again welcome Dr. Cody Haun back onto the podcast. How are you doing today, Cody?
I'm doing very well, David. How are you today?
I am not too bad at all. I'm quite good. I'm fantastic today, if I'm being honest. So it's been about a year since you've been on the podcast, and maybe things have changed up a little bit for you in that year. So if you'd like to bring people up to date with kind of your current roles, what you're up to at the moment, and where your research focus is at the moment.
Sure, man. I accepted an assistant professor position at Grange College right after our podcast last year. So I've been here for almost a year now, teaching some really cool classes. We offer an undergraduate degree in exercise science and then a master's of science degree in strength and conditioning and a master's of arts online in strength and conditioning. And that program is growing and having a lot of fun helping build some of those classes and rewrite the curriculum to focus really on what our students desire. That's been a really fun experience. And it's very performance focused, given the name, right? Strength and conditioning. So we're having fun with that. And I've been also serving as the sports science coordinator. So doing a good bit of athlete monitoring to help optimize the training of our athletes here. And a perk of the program as a master's student is you get to work with the teams, so the athletes here, and we help mentor our graduate students as they coach, and we assist in coaching, but primarily in a more of a mentor role. So we want to give them that hands-on experience. And like I said, we're doing a good bit of athlete monitoring, trying to enhance the athlete monitoring that we provide here as well to hopefully win games, right? So, and keep athletes healthy. So, yeah, man, it's been a really good time. And last time we talked, I was fresh off of my data collection, and I've been working with my colleagues and my mentor from Auburn University, my PhD mentor, Dr. Mike Roberts, on publishing all of that data. We've been working hard on that now for about a year and found some pretty cool stuff, I think. It's interesting, a lot more work to do. More studies to do, more research to do, but that's been fun. Been working primarily on that and getting research projects started here at LaGrange that we have planned for this summer and fall. So I feel really blessed and fortunate to be here, and it's been a really good year, man. How about you?
Yeah, it's brilliant. Ever since I had you on last time, I've been doing 40 sets a week on every muscle group and getting really jacked.
Yeah, 40, huh? That's aggressive.
Yeah, but again, and that was, if people don't remember, the high volume study which we discussed last time, and some very interesting data coming out of that. As you said, you're starting to publish some of that, and we'll get into that today. And what kind of the focus I wanted for this episode was to start off maybe in the more kind of hardcore deep science stuff, talking a bit around mechanism, hypertrophy and different types of hypertrophy, but then to bring it out and maybe show the implications of this in an applied setting, and implications that for an actual coach on the ground and what that might mean. And then, as you said, data collection and athlete monitoring is a huge thing you're doing at the moment. So I think it'd be nice then to discuss that at the end and kind of dispel some of the misconceptions around data collection and interpretation within sports sciences. So in terms to kick it off and to get quite technical, is we have some of your recent publications looking at differential types of muscle growth, namely sarcoplasmic versus myofibrillar muscle growth. And anyone that's been around the industry would have heard these terms come up and be debated and misinterpreted probably and misextrapolated over the last couple of decades. So to give people a bit of context, maybe Cody, you'd like to start off by maybe just defining what we mean when we say sarcoplasmic and myofibrillar hypertrophy and kind of the differences between those two concepts of muscle hypertrophy.
Absolutely. So the general definition of hypertrophy that's commonly used is an increase in muscle size. And this is assessed in a variety of ways. We published a review article in Frontiers in Physiology discussing all of the different methods used to evaluate if hypertrophy occurred and to what extent. And so to just sort of simplify first, hypertrophy can be considered as an increase in a muscle size. All right. Simple enough, right? So we have, I think analogies might help. Hopefully it's not more confusing, but I've started to use more analogies to make sense of some of this data because it can be a little confusing given the dimensionality of measurements and all the different things that can go into changing a muscle's composition and size. So if you think of it like a house, hypertrophy would just be an increase in the house's size, right? But that doesn't tell you what materials were used to make the house larger, right? It doesn't tell you the dimensionality of the addition of, to the house or the renovation of the house, right? It's simply the house is larger. So if we kind of zoom into hypertrophy and think of it as either myofibrillar or sarcoplasmic, and in the review we also argue that connective tissue hypertrophy, or you could think of it as like the extracellular matrix of skeletal muscle as a tissue, could also enlarge, right? And that could contribute to an observed increase in a muscle size. So we could add connective tissue hypertrophy on there as well. But if we just stick to myofibrillar or sarcoplasmic, it's first important to start with a muscle's composition. So a muscle fiber is this cylindrical cell, right, that consists primarily of myofibrils, so somewhere around 70%. If we were just to sample a muscle fiber and sort of plot, right, the data or understand the percentage of that fiber that is myofibrils, which are sort of just like mini muscle fibers that run the length of the muscle fiber. These are also these cylindrical thread-like structures that run the length of the fiber. Those are myofibrils, right? And those structures are generally going to occupy about 70% of a muscle cell.
Yeah.
And the sarcoplasm, so the fluid of the muscle cell, is obviously important to consider relative to its size, right? So let me back up for a second. The other two constituents that occupy we're talking more from a structural standpoint, right? Not necessarily like the absolute percentage of the fiber itself. So when we're talking from either like sarcoplasmic reticulum or mitochondria or the membrane or myofibrils, which are really the four key structures that contribute to a muscle size, regardless of fluid, myofibrils occupy the majority of that composition. Does that make sense?
Yeah, 100%.
So of those structures, about 70% myofibrils. And then, like I said, the sarcoplasmic reticulum, which is related to the T-tubules, right, the transverse tubules, which are involved in the propagation of an action potential and the release of calcium so that eventually muscles can contract, right? And you can tell me if I need to back up or if this is too elementary or something, man. I'm happy to simplify more or talk more complicated. You just let me know what you think would be best for the listeners.
Yeah.
But the sarcoplasmic reticulum and the T-tubules really are mostly connected to the membrane. So it's sort of a network, right? Because the action potential is propagated sort of across the membrane and then traverses the fiber, right, which eventually allows calcium release to occur. So the sarcoplasmic reticulum and the T-tubules, and then the mitochondria occupy most of the rest from a structural standpoint, right? And we're not talking about fluid. Like weight of muscle is about 70% fluid.
Yeah.
A little bit more or a little bit less, depending on various contexts. But does all that make sense so far?
Yeah, it does indeed, Cody.
Okay, excellent. So myofibrillar hypertrophy simply indicates that either an increase in myofibrillar number or myofibril size contributed to an increase in a fiber's size. And then sarcoplasmic hypertrophy simply means that, and we offered a more formal definition in the review, but a way to simplify it is an expansion in the sarcoplasm or potentially some of those other structures that could contribute to an increase in a muscle fiber size that doesn't involve the myofibrils.
Okay.
And, some of this is semantic, right? Like, does it really matter? And that's sort of where we want to get to today. Like, what's the practical application of this information? But going back to the house analogy, if you're building a house and you want to increase the size of the house, an important consideration to make your money be spent wisely is to consider what the house is going to be made of, right? Like what materials are you going to use? Are you going to make an addition to the house that's actually functional and that you'll actually use? And will the portion of the house that you're adding be strong? And will it be able to withstand weather, et cetera? So when we consider an increase in muscle fiber size, I think it's really important to consider what contributed to that fiber size, because that will affect its function. If we see, and so how we typically measure this in a laboratory setting, at least how we've measured it at Auburn and a lot of other labs attack this question, is you take a biopsy, and when we say an increased fiber cross-sectional area, we are referring to immunohistochemical staining. And so we've taken a biopsy, we've cut it into these thin slices, we've placed that slice on a microscope slide, we've stained it with antibodies that allow us to identify the membrane, and then we'll use a microscope to measure the cross-sectional area of the fibers in that cross-section, and it's common that we'll measure about 100 per cross-section at any given time point. And we'll measure the cross-sectional area of those fibers. If, let's say, from pre to 8 weeks post, we see an increase in cross-sectional area of those fibers, we'll conclude that hypertrophy occurred, right? Going back to that general definition, an increase in muscle size occurred. Yeah. But that doesn't tell you what contributed to the increase in the muscle size. Was it fluid? Was it proteins within the fluid? Was it myofibrils? Do we have more myofibrils, or are the myofibrils now larger? So that's why we've really been trying to assess that area and figure out, okay, we did see an increase in fiber cross-sectional area in this subset of the participants in my dissertation, and we're trying to understand the mode through which hypertrophy occurred, like what contributed to the increase in muscle size. So does that make sense? Did I clearly define like the difference between myofibrillar and sarcoplasmic hypertrophy?
Yeah, I think so. And the analogy was quite nice. So I think people kind of get the idea that at a very fundamental level that it's looking to see whether the increased size is down to increase in fluids or actual proteins within the muscle, and obviously all the caveats that you went into around there. And I suppose within kind of the literature or the industry to date, what has been the general consensus? Like, what have people believed to be the case of the relative contribution of each of those modalities of hypertrophy to overall increases in muscle size?
Yeah, excellent question. And so the general consensus is that if we see an increase in fiber cross-sectional area, the cell grew in a proportional manner. So those compositions that I discussed previously, right, mostly myofibrils, fluid, and the sarcoplasmic reticulum, the T-tubules, and the mitochondria. If we were to literally have a bar graph and we had baseline percentages of each of those constituents of the cell, the thought is that they increase in proportion, like their relative contribution to cell size remains the same, but the absolute size of the cell is now different, right? Yeah. And what we started to see is that wasn't the case, at least based on our analyses, that the increase in cell size wasn't consistently proportional to what we would expect. So that is to say that potentially hypertrophy can occur through the expansion of the fluid in the cell, of proteins in the sarcoplasm, for example, like glycolytic enzymes, other things, and the sarcoplasmic reticulum, the T-tubules, right, mitochondria. We think that's a reticulum now, right, where it's not those bean-shaped organelles just floating around. But all of those things might increase, including the membrane, right, the membrane volume. And so all of those things could hypothetically increase with a maintenance in myofibril number or even myofibril size. So is that making sense?
Yeah, it is.
Excellent. Yeah, so I think it gets back to really a simple question, which is the change in muscle fiber size, I guess a better way to ask that question is the proportion of a muscle fiber before an intervention preserved as it increases in size, regardless of training intervention? And my thought is that, revisiting a classic principle training from my practitioner side, specificity, right? There are specific adaptations to impose demands. It makes sense to me that the cell would respond in the cells, right? The muscle fibers that are involved in the training will respond in a very characteristic way, right? And then in response to higher intensity, perhaps like 85% 1RM or more with lower rep per set values, 2 to 5 reps, for example, and with lower volumes, it makes sense that the cell would respond in a very specific way to that, right? And that's what I was getting at with Dr. Stone's, some of his original arguments with block periodization and how we sequence various training phases to eventually enhance the power output of strength power athletes, but we don't just only exclusively train high velocity movements all of the time. And one of the reasons beyond, neurological adaptations and various skeletal adaptations and connective tissue adaptations is the protein level changes in muscle. So to your point, like, is hypertrophy functional? And if so, like, what type of hypertrophy is quote unquote functional? And obviously that gets to what do we mean by functional? So I think if we're talking about strength, which is the most common assumed relationship, right? If a muscle fiber grows, then it will have a greater capacity to produce force. And I think that's logical. But we recently published a commentary discussing how it may not be that simple, right? That there are examples of hypertrophy occurring where you don't necessarily see an increase in strength. And myofibrillar hypertrophy specifically is likely going to contribute to force production capabilities more so than other types of hypertrophy by comparison. So I guess the answer to the question is that the relationship between hypertrophy and the desired performance outcome depends on the type of hypertrophy, one, how hypertrophy is measured, two, and then three, really what is the desired performance outcome? Is it strength? Is it power? Is it endurance? For example, like in my dissertation, for example, we had them complete 10 reps per set on average, right? And that happened in very few cases, a little less, but most of them were able to achieve the exact program training structure. That was 10 reps per set. And we started with a lower, so it was like, making me go back to the first week of the study. We started with 10, and then we worked all the way up to 32 sets of 10. Well, the overall study design, most people would characterize as relatively light loads and high volumes. Right, so we worked all the way up to where these guys were doing 32 sets of 10. That's a lot for a movement in a week's time at 60% 1 rep max. So relatively light loads, high volumes. And based on the analysis that we've performed so far in a subset of the participants where we observed an increase in fiber cross-sectional area, we observed on average a decrease in the concentration of myosin and actin, so the contractile proteins, and an increase in sarcoplasmic protein concentrations based on the assays that we've done so far. And, this is all in review. And of course, it's being critiqued heavily, which it should be, right? It's a relatively new finding. And so just to pause there for a second, we want to know the truth. Like we were talking about before the call, like it's not like you can go get $1 million to do these studies. There's no real obvious financial incentive for us to publish this paper, right? So we want people to critique it. We want to know what the actual truth of the matter is. We don't, want to be the group that discovered sarcoplasmic hypertrophy to pad our pockets. Like that's not happening, right? So just to be clear on that, like I'm really glad that people are offering critiques and we want to know, is this good data? Like, is this actually what happened? And we've certainly tried to do our best, in the lab, and we were very careful with these analyses and didn't expect to find what we did. I'll be the first to tell you I didn't expect to find what we did. And we ran multiple assays and ran them in different ways just to try to make sure that it was correct before we published it. And we found that the sarcoplasmic protein concentration increased, and that those contractile proteins, based on the technique that we used, which involved a combination of purifying the sample to extract the myofibrillar fraction, and then studying that portion of the fiber, we used a protein blot, basically, to simplify the methods. And you can see, with the technique with it that we used, you can see the density of myosin and actin bands, and we found that the concentration, so when you normalize the density of the the band to the input muscle weight, we found that the concentration of those contractile proteins decreased relative to the change in muscle fiber cross-sectional area. So just to kind of zoom out for a second, the cross-sectional area of the fibers in that microscopic image increased, whereas the contractile protein concentration appears to have decreased, while the sarcoplasmic protein concentration seems to have increased. So considering that, that's what we refer to as sarcoplasmic hypertrophy, right?
Yeah.
And if you think of the training design, it was the glycolytic metabolism was, well, I guess a good way to say this is, one, ATP turnover was quite high, right? Like those fibers were contracting a lot in a week's time. So the ATP demands were relatively high. So to kind of revisit the specificity comment, it makes sense that, especially given the amount of reps per set, so each rep taking about 30 seconds, going to our bioenergetics knowledge on energy systems, we know that the glycolytic energy system is going to be the primary energy system that's involved with providing ATP so that those muscles can continue to contract, right, during the sets. And we did see an increase using proteomics. We saw an increase in various glycolytic enzymes in the sarcoplasm, right? So that's an example of specific adaptation to an imposed demand. And other proteins that make sense. And I think that once we study and use similar assays, that once we study a higher intensity, lower volume paradigm, which we have scheduled for this year, my good friend Chris Van is going to do a really nice dissertation looking at different training structures and their effects on the ultrastructure and the protein level response to those interventions. So we'll be able to see if a different training configuration that is a higher intensity and a lower volume approach results in a different adaptation. Perhaps in that context, considering the relative force demands of that style of training and the forces involved, right, all of those tensile forces in the fibers, it makes sense that the sarcomeric proteins and the contractile proteins and those costameric proteins and proteins involved with force transduction specifically, it makes sense that those would be better maintained or at least increased. Or I guess at least maintained, but perhaps increased in abundance. So, yeah, with all that said, man, I really think that this follows logic that's really centuries old, really, and we're just now able to probe the tissues at a deeper level, and we're finding that indeed it does seem that there are specific adaptations to imposed demands, right? And I think a combination of proteomics and transcriptomics going forward, where we can understand things at the mRNA and the protein level at a deeper level, like those are going to be necessary to really tease out, if you do this style of training, what is the resultant response to that training at the fiber level. And then we need to really isolate single fibers and look at the single fiber alterations in force production and power output and you know what I'm saying?
100%. And I think, say from my own personal perspective, someone who may not be as versed or an expert in muscle physiology as someone like yourself, Cody, when I'm listening to that, it makes sense when put up against the body of literature where we've now good literature to support that hypertrophy can occur at all percentages of 1RM, and we see that studies showing hypertrophy coming at 30, as low as 30% 1RM, and similar to 80-90% 1RM when matched for proximity to failure. And again, this could be the prevailing idea that yes, you can induce hypertrophy at those lower levels of intensity, but the muscle function outcome, and which we have seen in the literature, that yes, the hypertrophy may be similar, but the strength outcomes is not nearly the same, where the rationales being put forward in the past have been, it's lack of specificity, they haven't trained strength, where it might be actually something a bit more in the muscle physiology itself. It might be that these lower percent 1RMs, this pump work per se, causes this sarcoplasmic hypertrophy, which, as you say, when we think fundamentally of basic age-old training principle of specificity, makes sense. You're putting a different metabolic demand upon the muscle, and the muscle is going to respond, adapt to that demand. And I suppose that segues into your commentary in a certain way of the role of muscle hypertrophy in muscular strength gains. And could this be one of the reasons why we don't always see a nice linear correlation between increases in muscle hypertrophy and increases in muscle strength?
Really insightful comments, man. So one, I did want to comment on when you match for volume, you see similar hypertrophic outcomes to light versus heavy loads. Sort of that comment, which is right, that's spread all over the internet now, right? Like, it doesn't make a difference as long as you equate for volume. Light loads result in the same adaptive response or hypertrophic response to heavy loads. Well, what's really important about that data is very few people have assessed the myofibrillar fraction of the tissue, right? And a lot of those techniques have been ultrasound based, or DEXA, like whole body x-rays, different techniques like that, which, hey, those are useful. Like we said in the review, like, do the science. That's great. But we just want to be really careful about these broad sweeping conclusive claims. I don't think that the long-term hypertrophic outcomes and the ultra-structural and morphological adaptations to light resistance training are going to be the same to heavy load resistance training. And I'm talking on the sort of the ends of the spectrum, right? Like 30% or so 1RM versus 80% 1RM. I just don't think that the protein level adaptations are going to be the exact same. I think there are going to be very characteristic responses and ultra-structural responses to those styles of training. And if not like in muscle alone, we need to consider bone, right, and connective tissue and the nervous system. Like, what do you want to hypertrophy for? What is the eventual training outcome you're trying to achieve? So with that said, to your point, I do think that sometimes in some of the studies that we have seen that have shown little relationship between quote unquote hypertrophy and strength relate to the fact that the hypertrophy that occurred could have been mostly sarcoplasmic. Right? It probably didn't involve a significant increase in myofibrillar proteins, which are involved, right, in direct force production. Myosin and actin binding, forming a cross bridge, and then eventually force transduction to the bone, and therefore measurable changes or differences in force production. But anyways, yeah, I do think that that's one potential reason why we don't always see an increase in strength due to increased hypertrophy. Now, as you have read the commentary, I think that's just one point. And just to be fair, I don't want to mischaracterize Dr. Lineky and colleagues. I don't want to mischaracterize their views. I have a lot of respect for Dr. Lineky. I think he's made us all think critically about our ideas in the field. And I was actually going to do my PhD with him at Ole Miss before I went to Auburn. So I have a lot of respect for him. And it was fun to do the commentary. But we don't agree with their points, and we think that our arguments are better. But that's cool, right? Because it's science, man. Science. Like, as long as we all want the truth and we want to do more research, we can discuss things. And that's my goal with this whole thing, man. Like, if I wanted to get rich, I wouldn't be doing this.
No one goes into research to get rich.
Right, exactly. So it's like that. I love the process. I love the research side of it, and discovery is like a drug to me. I love science. I love learning, and I think this is a good discussion. But I do think that that's only one point of the many, and I can go into some more of those if you would like, or do you want to ask any more questions related to that or clarify anything?
Yeah, no, I like your clarification there, and it was something I should have brought up. As you said, these studies that have shown, yes, and you said you can't go on the internet without people like, oh, you don't have to lift heavy, or you lift light loads, go close to failure, you get the same result, which, as you said, all these studies are mainly just looking at cross-sectional area. And if you give an analogy, if you said to these same people, oh, well, I did this training plan and all groups went from 80 kilos to 90 kilos on the scale weight, therefore they're all equal, they wouldn't accept that at face value. Like, oh, well, it kind of matters whether, do you have DEXA results to show whether that was muscle or fat that they gained?
Exactly.
Like, it doesn't matter, they all gained the same. Look, the mass went up the same. It's like,
great point. Yeah, that's a really good point, man. I'm going to steal that one from you if you don't mind.
You are more than welcome. Where when we look at these studies critically in hypertrophy, maybe that's the next stage, that we need to combine cross-sectional area with myofilament protein content then as well, to really get a contextualized story of what's going on there. And go ahead, Cody.
Also, just to be clear, this is difficult to measure. It's difficult to measure. I repeat, this is not easy. It takes so much time. Like, the amount of work that goes into extracting the tissue from a real human, right? Well, one, convincing them to be in the study and having them perform a training protocol under supervision, harvesting the tissue carefully and painstakingly, caring for that tissue like a small child, and like all of the hours that go into meticulous weighing of samples, making solutions, carefully pipetting the exact volumes. Like, I have so much of an appreciation for any lab that has measured like protein concentration in the muscle sample and pulled that off in context of a training setting. That's why we don't see it much. It's difficult and it's really expensive.
Yeah.
And so I just want to be clear on that. Like, and we're trying. We really want to keep probing this area, but it costs money and it takes a lot of time. And I just want to say, like, that it's not to say that the other authors or people that have investigated this area and used ultrasound or DEXA or some other technique are doing it wrong. I don't feel that way. But to your point, if we really want to understand the protein level changes, right, the ultrastructural changes, we have to extract human tissue. And it's just not easy to do. As long as we appreciate that a lot of those studies haven't extracted muscle tissue and measured protein level changes, and we interpret their data with that limitation, that data is still really useful. Anyways, sorry to interrupt you there. I just wanted to make that that really clear.
No, it's 100%. I think you've turned a lot of people off the idea of research. Maybe people who are considering an avenue of research, you had some kind of Vietnam flashback there of separating tissue cells.
I did flashback and started like itching my neck a little bit. No, it's fun in a really weird way, right? Like, for me, it's a means to an end, of course, but as long as you keep your eyes on the prize, a lot of that meticulous, slow lab work, you can get through it because you know that the ultimate outcome, as long as you're really careful and you're taking your time, and there are long days. I mean, just for a few samples, there are sometimes, and dude, that's if it goes right. You know what I'm saying? Like, you do Western blots. I can't tell you how many times I wanted to throw our lab equipment out of the window at Auburn. You run a Western blot, which takes basically all day. Then it has to rock overnight with an antibody, and then you come in the next day and you try to read your blot and you don't see anything. It's very frustrating. But as long as you keep your eyes on the prize and you focus on the purpose, right? Yeah. The purpose of it all. And you give yourself time. I think that's where, like with lab work, a lot of times people get really frustrated and kind of get burnt out. I think one of the best pieces of advice I can give is it's okay to take your time, right? It's okay to take it a day at a time, do what's realistic, and focus on the purpose of the assay, right? Like, what question are you going to answer? And understand that this technique is a step forward, right? It's getting you closer to answering that question. It can help make those longer days, a little easier on you. But yeah, anyways.
No, that's 100% true, and I can relate to all that myself. And as you said, it's like any craft. It can be meticulous at times, but it's a means to an end. And I suppose in answering those questions around the role of hypertrophy in strength, we'll kind of jump forward a bit, because I think most listeners and everyone can accept that, it's not always a perfect linear relationship between if you get bigger or you hypertrophy, you get stronger. It doesn't always work that way. And conversely, a lot of people can get stronger without gaining any muscle size, without getting any bigger. So I suppose the real question, Cody, is to what extent and under what circumstances does muscle hypertrophy contribute to strength gain?
Yeah, that's like you are quoting our commentary, right?
Yeah.
That to us is the more interesting question, because as long as we appreciate the nature of causality and that so if we sort of just clarify what I'm talking about, a necessary cause and a sufficient cause are different than a contributory cause. And in the commentary, we argue that hypertrophy isn't necessary or sufficient to increase strength, but we feel strongly that it's a contributory cause, right? And so a necessary cause is basically if not A, then not B, right? Sufficient is if A, then B. And we know that, like you said, hypertrophy can occur, and there are examples in the literature where hypertrophy measurably occurred, but we didn't see a significant change in strength. And we see that anecdotally from people in the short term. Concede that it's not necessary. You can get stronger without significant changes in muscle size. But there is plenty of data, we feel, both theoretical and practical, that shows that hypertrophy is a contributory cause, and particularly in context of someone who's trained, who's passed those initial neurological adaptations, not a necessary or sufficient cause. And there are quite a few examples in the literature that we could use, and I would encourage anyone who's interested to read the commentary, because I'm simplifying a lot of deeper thought on the matter. But, for example, there's a study that it didn't even make it into the commentary that I use in my classes as an example of how myofibrillar hypertrophy contributes to strength, and it's from Paul Cribb in 2008, and they measured myofibrillar protein concentration and showed a really strong correlation with increases in strength. Like basically the more myofibrillar protein concentration increased, the larger the increase that was observed in strength. So to dismiss that and say that it's correlating random error, which we feel by definition means that like hypertrophy and strength, there's no obvious reason why they should randomly correlate with one another. We think that that's evidence, and there are more examples. We think that that's sufficient evidence to conclude that hypertrophy is a contributory cause. And I mean, there are other examples, but we were cutting short on time, I know, and I've got to go teach advanced sport nutrition. So
that is a rabbit hole that we could spend hours going down and still probably come out with no conclusive answers. And I suppose with all these interesting unanswered questions, where do you see the future of this research in kind of muscle hypertrophy, this muscle physiology area? What do you see as what excites you going forward? What are the exciting questions that you think we need to get answered?
Yeah, so I tell people I'm a scientist, a coach, and a professor. So for me, I love the heavy muscle phys. I really enjoy it, understanding things at that level. But I want to translate that work into practice. And for me, the studies that I mentioned previously, where we dose training and we have a really nice, well-controlled training study, and we measure things like I mentioned, proteomics and transcriptomics. So a more complete mRNA and proteomic or protein level characterization of the effects of specific types of training. And that is like, if you do this style of training in this set of the population, in this context, what are the specific protein level adaptations? And a really complete examination of all of the known proteins in muscle. And then taking that information and looking at function. Like, okay, we see this observed change in the proteins in muscle. What's the change in function? In other words, like, so what? Like, does it matter? Does it improve jump height? Does it improve, you get it. I see you shaking your head. So, like that's really exciting to me. And I think that's the direction that we have to try and move toward, and taking some of this deeper muscle fit stuff and ensuring that we're, at least for me, I want to always aim to apply it, right? And make it useful for people, to help people achieve their goals and be healthier and live, more fulfilled lives. So.
Yeah, and I think that is important, and we should never forget that. I know that would be a passion that you and I would both see, that we like this deep kind of hardcore science end of it, but ultimately the end point should be to make better athletes, healthier humans, and more successful human beings all around. And I suppose that is one reason why you've been involved, to kind of close off with a very exciting company, I should say, Aplyft, at the moment. I've been lucky enough to kind of have a glimpse behind the curtain of what's going on there. So for people that may not be familiar with Aplyft, maybe you'd like to finish up, Cody, by just giving them a brief overview of what the ethos and mission of that company is, and why you're involved with them.
Sure, yeah, definitely, man. So to keep it short, because like I said, I only have a couple minutes and I have to go teach. There's a really nice website that goes into a good bit of detail about Aplyft and the mission, and people can find all that information at aplyft.com. That's aplyft.com. But man, I've been online coaching for 6 years total, and throughout that time, I have used tons of different workflows: Excel, Google Sheets, Docs, WhatsApp, Facebook Messenger, Skype, etc. Right? And PayPal, Stripe, different methods. Vimeo, like it goes on and on. And I've wanted consistently, and one of the earliest problems I dealt with was data entry. Like I've always tried to be data driven, or at least steered, right? And use the information data we can collect from clients and better program their training. And one of the roadblocks to that process is the client actually entering the data that we want. And using a spreadsheet system or other workflows, I found over the years, like sometimes it's not the most convenient method for someone to enter data, and it makes it less likely that they actually will. So I started looking, for different apps and workflows that might work, and the guys at Aplyft contacted me, and we started talking, and it turned into a really cool relationship, and I have a lot of confidence that they're good people, and the mission is essentially to centralize, and that's really what the portal and the app does. It increases the likelihood that clients that you might be coaching remotely will actually enter data, because it's a mobile application, right? And most people are on their phones a lot nowadays. So it increases the likelihood that individuals will actually enter their data for us as coaches. But for the coach, from my perspective, it's really nice to have everything centralized, right? So video, chatting, audio, pictures, all in one place. All the payments are processed through the app, through Stripe. And the portal, it's actually undergoing significant renovations, and I'm really excited about the changes, because I've helped for the past year just to consult on different changes that would make online coaches' lives easier and provide the information in a really efficient manner, and get everything in one place for the online coach to see things like body weight data, nutrition data, and of course the training related data all in one place. right? And ensure that it's quickly viewable, it's useful, and it can influence the training decision, right? It's not in 8 different tabs in 4 different softwares, etc. So centralizing everything was a real draw for me. And the more it's progressed along, like, people are interested, and other coaches like yourself dealt with similar issues and were looking for a similar service. And my goal is to help provide a solution to that, helping Aplyft provide that service. So that's really the overview, just this centralized software, mobile application, and coaching portal that helps improve the experience from an online coach and then a client that they're working with remotely. And as I said, I'm excited about the changes to come. It's been a great experience working with them, and they're actually sponsoring, IPF World Championships, and I think also Raw Nationals. So this year, so a lot of cool things ahead, man. I'm really excited.
100%, and really looking forward to seeing what the guys come up with over at Aplyft there. So I know you're in a rush, Cody, so we'll finish up now. If people are looking to follow you or get in touch, where's the best places to find you?
Sure, man. I'm on Twitter, Instagram, and Facebook now. So now that I have a life outside of, I'm kidding, a little bit. As a professor now, I want to help put out good information. I'm not a huge fan of being on my phone and on social media, but I'm sincerely trying to put out good information here and there when I'm not teaching or doing research. So I am putting stuff out on Twitter, Instagram. I occasionally will put out some information related to training and nutrition and stuff like that. And then I also have Facebook, but and on Aplyft for coaching. And then I'm also listed as a coach on Greg Knuckles' Stronger by Science site. So I'm online if you search for me. And then in terms of research, I'm on ResearchGate is where I update my profile, my research profile, and try to keep things updated there so people can access my papers and find my work there scientifically. So, yeah, man, I got to run. It's been a pleasure. Thanks for having me on, and maybe we can get on together soon and get on another call and talk about some of the more nuanced stuff with hypertrophy and strength, because I know we had to kind of cut that a little short. 100%.
I look forward to it. Thanks so much for everything today, Cody. Absolute pleasure.
All right. See you, buddy.
There you have it. Episode 82 with Dr. Cody Haun. Now, I hope your mind isn't fried too much after listening to that. I know we got into some deep science things there, but I think it brings up a great point, and Cody then embodies it as a researcher, that we are looking for the truth. Whatever we do in research or as coaches, when we collect data and we use data, we're always looking for the truth, and we want people to critique, and we want external criticisms and views that allow us to get to a closer estimation of what we believe is fact or is the truth. And within that, we are only as good as our measures. We can see that upon Cody's research, this implications of measuring the myofibrillar protein content within muscle hypertrophy, along with increases in size and cross-sectional area, may lead to us viewing differently the results of previous studies, and it may have implications for what we extrapolate from such studies. And the same thing goes for what we do with our data collection for athletes, because we're only as good as our measurements and the accuracy and sensitivity of our measurements. 20, 30, 40 years ago, we didn't have the same degree of technology that we could go down and examine the myofibrillar protein content with such accuracy and make these implications and make these extrapolations. So we just had to go with what measurements we had. And the same goes with our coaching systems, our athlete development systems. We only can go on the measurements we have, but we have to ensure that the measures we are using, we understand the limitations of them, we understand the reliability and sensitivity and validity of those measurements. Because if we start putting too much faith and we allow the data to make the decision for us rather than letting the data guide our decision making as coaches, we can get down a slippery slope there, where if it was just a case that it was simple as collect data measurements and they make the decisions, we could set up a computer algorithm that does that all. But then coaching is far more complex than that. Human beings and athletes are far more complex systems than simply boiling it down to ones and zeros. So I think that's just something to always keep in mind. In a world where technology is always advancing and progressing, and as coaches we can kind of get distracted by the new shiny thing, that we shouldn't let the art of coaching go, and we shouldn't hand over all of our autonomy as coaches to computer systems, as we can sometimes see that happen with GPS systems, fatigue monitoring systems, HRV, all these kind of things, where we take our own brain and our own ability to think rationally and logically out of the situation and just allow these systems to guide us, which can be a slippery slope, as I said. Anyway, that's a rabbit hole I'm starting to go down into. I'll leave it there. Make sure you subscribe to the podcast. If you enjoyed it, share it with someone. And on to next week. Have a great one, guys.